here. I guess I'll say a few words about myself and where I'm coming from. So I'm in the Department of Computational and Systems Biology at the University of Pittsburgh. We're in the School of Medicine, and the department has been in existence since 2005. We also have a graduate program in computational biology, of which you have a distinguished member in your presence. So you should all know Sanjana by now, hopefully. If not, talk to her. She's doing interesting things. And so today, I'll talk to you about rule-based modeling approaches, which is something that my group has been active in developing, with the ultimate goal of large-scale modeling of biochemical networks, which I think is going to be of interest to you here at the workshop. So modeling cell regulatory networks. So here's a cell with lots of receptors and lots of signaling pathways connecting the different processes within inside the cell. There's cell cycle regulation in here, and those are, of course, all connected to the various regulatory components. And ultimately, we would like to have, if you want to make a whole cell model, you've got to consider all of these signaling that's taking place. Our current understanding tends to be focused on one system or so at a time. So we have models of various pathways, and I'm sure many of you are familiar with such models, where we describe the biochemical events in detail. And ultimately, we'd like to be able to link these together, all of these different modules together, and to make unified models that would predict how a cell would respond to multivariate inputs. So that's the challenge. And I think any of you who have done some modeling of these kinds of systems appreciate the scale of that challenge. So I'm going to focus on four key challenges that are addressed by some of the techniques that my group has developed in the last 10 years or so. This is not an exhaustive list of challenges, and I'm sure that at the workshop you'll cover many different things in addition to this. But the four challenges that I lay out for modeling cell regulatory networks, the problem of just specifying what the model is, and we'll see how that problem arises from the complex molecular biochemistry that has to be considered in such models. We've developed rule-based modeling languages. My group develops a tool called BioNetGen that you'll learn about in the lab today to be able to specify such models in a precise and efficient way. And so we can write down models of large cell regulatory networks. That gives rise to another challenge, which is how do we efficiently simulate such models. So we can, in the earlier days, we were able to write down models that we simply couldn't simulate with the tools that we had at the time, and we had to develop new tools to be able to simulate those systems. And then so this describes capabilities. The first two parts describes capabilities that are being developed for rule-based modeling. And the second two are going to talk mostly about how do we take existing models and allow people to see what's in them and to compare different models. I think there's already been some discussion at the workshop about how to compare different models of the same system. And I'll talk about a tool that we've developed that I think will facilitate such analyses, and then how we can visualize these large-scale regulatory models in a zoomable way that lets us get a global view of the cell regulation and then zoom down into the details, how different aspects of the biochemistry is being modeled to allow us to compare different models and to understand what's been described and what assumptions have been made. So we'll span a different range here. So we're going to go back in time a little bit, first to talk about model specification and the development of rule-based languages. So the basic problem that rule-based modeling deals with is summarized in this slide, and that is that problem of combinatorial complexity, the combinatorial explosion that occurs when we consider molecular interactions and their implications. So on the left-hand side, I've shown one way to visualize the components of a cell regulatory network in terms of a contact map. So this shows different proteins that are involved in signaling. Many of those would be familiar to people who study epidermal growth factor receptor signaling, for example. There are post-translational modification sites, tyrosines, that get modified when the receptor dimerizes. These sites serve as binding sites. The phosphorylated sites serve as binding sites for various adapter molecules that recruit then downstream effector molecules to this receptor complex. And the basic molecular interactions that give rise processes are fairly straightforward to write down in a simple way, and you can see that there's a relatively small number of them. However, if we, in an unbiased way, write down the implications, what different molecular complexes can form out of these interactions, we do this combinatorial enumeration, and we start to arrive at rather large and uncomfortable numbers if we're trying to code these equations, for example, into a computer and simulate them as differential equations. So code the models, that is. So we can see that, for example, there are 3 different sites on the receptor, and we can enumerate the possibilities at each site. The receptor, this ligand binding site of the receptor can either be unbound or bound to an EGF molecule. The tyrosine site 1068 of the receptor can either be unphosphorylated, phosphorylated. If it's phosphorylated, it can be bound to Grab2. If it's bound to Grab2, it can be bound to SOS and so forth. So we have a chain here. And on the second site, we have a different chain. There's an adapter molecule called SHIC, which itself can become phosphorylated and bind Grab2 and SOS. And if those of you familiar with EGFR signaling, this is a very small subset of the interactions that can take place just at the receptor. So we get hundreds or thousands of different possible complexes coming from what's known about the biochemical interactions. And we have to deal with that in some way. We can sweep that complexity under the rug by making certain simplifying assumptions, or we can come up with ways to write down the models in terms of these relatively simple interactions and somehow let computers and computational algorithms deal with the complexity. And that's what we've chosen to do. So this is more background in terms of, this is really the lingua franca of signal transduction, looking at the domain structures of proteins, the sites of post-translational modification, how those proteins interact with each other to form signaling complexes. And people who study these systems are constantly trying to interrogate them to determine what functional role each of these components play in these networks. So the ultimately models of these systems have to be able to predict how, if we modify the structure or the function of a given domain, what other proteins it can bind to, whether it can be phosphorylated or modified in some other way, and how that percolates through the network. If we want to be able to make models to predict such things, then we have to make models that include these sites and how their implications go through the network. So we wind up having to deal with this problem of combinatorial complexity in one form or another. So the idea we can write down a simple model in terms of a relatively small set of interacting components. We can describe their interactions in terms of these rules, and I'll talk more about how rules work in a few subsequent slides. And then this combinatorial explosion that occurs, we have to deal with. Okay, so rule-based modeling languages were developed to deal with this problem, and the basic idea is that we simply abstract the structure of complex biomolecules in a simple way. What I'm showing here is a little snippet of BioNetGen language where we're defining a molecule type, which is this lin-tyrosine kinase. And we're going to describe several domains of the kinase, including SH3, SH2, kinase, and 2 tyrosine sites. And each of these sites can bind to cognate sites on other proteins, and they can also potentially have different modification states that would represent things such as phosphorylation. So we have a kinase, a lin tyrosine kinase that has 2 different docking domains, a kinase domain, and 2 different phosphorylation sites. And we can make rules then that describe how the interactions involving these different sites are regulated in the network. Another example of a system that we can describe in this structured but coarse-grained way is a typical immunoreceptor, the Fc-epsilon-R1 high-affinity receptor for IgE. Here we're representing its extracellular binding domain and 2 different tyrosines that are in different subunits of the protein. So there are multiple different ways we could have chosen to represent this multi-subunit protein. Here I'm representing each subunit as a distinct site in the molecule. I could have chosen to represent the subunits themselves as distinct molecules that are linked together in some way. So that even in the, there's already a coarse graining that's taking place at multiple levels, and their choices one makes in modeling. Yes, a question here.
Could you go back 2 slides, please?
2 slides.
So one biological possibility is that some of these states exclude other states. That
Absolutely.
When EGF is bound to the receptor, that it excludes some of the other proteins. Another possibility is that like maybe those 3 proteins at the bottom, that they aggregate so fast that you don't see a lot of the other possibilities.
Mm-hmm.
Has that kind of thing been studied? Does that happen? Because that would cut down on the space.
Definitely. We typically don't know whether such simplifying assumptions are correct or not. So our concept is to build models that where that full state space is possible, and then to use parameter estimation approaches, for example, to rule out, and by fitting to data, we can figure out which structures are possible and which aren't possible. It's difficult to directly measure them. We don't have a sorter that can sort the complexes by size and tell you, you've got these EGFR complexes with 2 receptors and 2 Schiks and 2 Grab2s. The evidence that we do have regarding multiple adapters, for example, binding to the same receptor, is that yes, you can bind, these are disordered regions of the peptide structure, the cytoplasmic domains of receptors, and you can almost certainly bind multiple adapter molecules simultaneously. So the stoichiometry of the complex is not highly restricted, and that's the typical assumption that's used to restrict the state space. Whether or not partially formed complexes are important kinetically is a question that's difficult to answer. But one thing that I can say, for example, if we have time at the end, and if not, I can show after, that, for example, the Schick phosphorylation here is competing with the receptor phosphorylation here for binding Grab2. And so you might think, well, this is arbitrarily complex, and we don't need to consider all that structure. But that competitive binding can actually have kinetic effects that have been observed in the system. So it's tricky to say which ones we can exclude and not exclude. We have also, and other groups as well, have tried to study the diversity of complexes that can form, and there's some interesting work in that area, which I can't concisely summarize at the moment. Okay, I think we covered what was here, so we can abstract the receptor. So what's a rule? And the key point is that we can translate typical statements that we can make about the biochemistry directly into rules. So we have here the Lin SH2 domain binds to phosphorylated tyrosine 218 on the beta subunit of Fc epsilon R1, and we can simply translate that into a rule that describes this interaction. So at this point, this part of the rule simply says what the interaction is. And we have typically, the syntax of rule-based languages has been, most of them is that you have chemical notation where you have reactants and products, and the interacting components are specified as the reactants, and then the product, the complex that's formed by that interaction is specified as the products. Here we have a bond being formed between the SH2 domain of Lin and the phosphorylated beta subunit of the receptor. The bond is indicated through this bang notation, where the index here refers to the index of a bond. It's just an arbitrary number that's used basically as a label so you can figure out where the endpoint of the bond is. So up to this point, we're just describing an interaction that can take place. The rate law then describes some phenomenological rate for this process. Typically, we assume mass action kinetics for these systems. So if we're going to instantiate this, if we're going to use this rule to instantiate a differential equation model, then the rates of this of the reactions that come from this rule are going to be described by law of mass action with these rate constants. A key principle here is that both Lin and the receptor have other domains, and the way that this rule is written is in the most general form. We're assuming that the states of the other domains do not affect the ability to form the bond here. So we're saying that as long as there's a free SH2 domain and there's a free phosphorylated beta tyrosine 218, this binding interaction can take place. Okay, so we haven't, that will happen regardless of the states of other components in these molecules. So this don't write, don't care is sort of a fundamental principle, and the upshot of that is that each rule in our network can potentially generate a large number of different reactions, and those reactions may generate new chemical species that build up the size of our network. But we don't have to enumerate in advance what those different possible species are. We just have to say what the possible interactions are. Okay, and yeah, so here this rule can generate many different reactions. In many instances, we use law of mass action for the kinetics of those reactions. So here the forward rate of this reaction would be this rate constant, KPL, the concentration of the particular Lin species that was involved in this reaction, the particular receptor species that was involved in this reaction. Okay, and you'll get to see in the lab examples of how this works out. So one then goes through a process of identifying the components and the molecules and their binding sites and post-translational modification sites that are going to be included in a model. These interactions are encoded in the form of rules. We can have a relatively small rule set. So our original IgE receptor model contained just these components, the receptor, the two kinases, and a cross-linking ligand, and about 20 or so rules, and generated 350 some different chemical species and more than 3,000 reactions. So in the BioNetGen file, the specification looks like this. This is the BioNetGen language. We have blocks to define our molecules and blocks to define the different reaction rules. And I'll go through a little bit of the mechanics in a few slides down the road. But this is a scalable, just to show that this can be a scalable procedure. I've just picked a diagram of one of the more complicated rule-based models that's been developed, describing early events in T-cell receptor signaling, and it was work that was done by my colleague Bill Hlavacek, his group, and published a few years ago. And this model has 20 different molecule types that are shown here, and this is a kind of an extended contact map describing in more detail the interactions that are in the model. There are 129 rules, and there's no possible way that this could be simulated as a set of ordinary differential equations, at least not that we know how to construct currently. So that poses a conundrum. How do we simulate such models?
So why do you say it can't be done with differential equations?
We don't know what the equations are. There are something of the order, I think about 10 million of them for this system. We could, I actually don't know, and they don't say in the paper. We would have to do the combinatorics here to find out exactly what the state space is. It depends somewhat on the assumptions, but I'm pretty sure that if you take this model and try to run it through BioNetGen, it will crash, take your computer down, filling up all of the memory before it terminates in network generation. So you really can't find all of the species if you want to try to enumerate the system in advance. You have to do something that doesn't involve finding all the differential equations and solving them. And so there are ways to do that, and that's the next topic. So, okay, so how do we simulate rule-based models? So up to about 2007, the procedure was we wrote down our model as a set of rules. We then performed iterative application of the rules to some seed set of species to generate a network. So there's a process of network generation that I haven't described in detail. We can go into it a little bit later, and you'll see that examples of it in the lab. So we build up this network. Once we have a network of reactions, we can simulate them as differential equations. We could also use the same set of reactions to do stochastic simulations. Those are based on knowing the reaction network. And then we use some kind of reaction network simulator. One could also do a spatially resolved simulation using PDEs, for example, based on the same reaction network. So it looks something like this in practice. Here's a contact map describing a scaffold protein interacting with 3 different binding partners, A, B, and C, there are here the interactions are assumed to be independent, so we have 3 different very simple rules, and each rule generates a set of reactions, all the possible binding interactions involving A, all the possible binding interactions involving B, and all the possible binding interactions involving C, and the different complexes that result from those interactions. And you can see that initially, if you start with, for example, the scaffold S, the A, B, and C molecules in the system, you would get the reaction S plus A gives SA, S plus B gives SB, and S plus C gives SC. If we now take these complexes, SA, SB, and SC, and see what happens when we add A, B, and C to them, we get the second reaction in each group. And then in each case, we then form a new species, SAB. This is an SAB. This gives SAC. SAC is new, so we get this reaction, and so on and so forth. So you can see that if we build this network up in an iterative process, we eventually build out all the possible species and reactions that can form. And so we started with 4 types of molecules and 3 rules, and we get 11 species and 12 reactions. Does that make sense to people? Okay. So it's pretty straightforward. We can, in tools like BioNetGen, you can actually control this network expansion. So maybe you wanted to only consider, for example, species that had up to a certain number of scaffold molecules or binding partners in them. You could limit the size of complexes that could form in the network generation step. The problem comes when we start to make contact maps models of our typical signal transduction systems that include sites that we know about from biochemical experiments. We can very easily write down a simple model, 5 proteins and 20 interactions that has 170,000 species. And that's beginning to that pretty much pushes us over the limit of what we can handle on a computer with a reasonable amount of memory. And the simulation time for a 170,000 differential equation model is pretty long, typically. So we don't really want to do that most of the time. The other problem is that if we add another site, we might easily go into millions here. So about 2007, a couple of groups, one that I was involved in, and a group involving Vincent Danos and Walter Fontana, who are the developers of another rule-based language called Kappa, came up with a way to simulate rule-based models in what we call a scalable way that didn't care about this, certainly the network size, and avoided network generation altogether. And what are shown here are the applications to some simple problems where polymerization occurs, so you get infinite networks. So if we go back to our simple example, the idea now is that instead of generating all the possibilities in advance, we have rules that are generators of events in our system. So here's how a given rule would generate possible events in a system of interacting molecules. So at a given time in our simulation, we have a box full of molecules of the type of the scaffold and the A, B, and C molecules, and they're in different states. So some of the molecules are freely floating in solution. Some of the scaffolds are bound to different components. And at a given time, each rule has to basically query or have set up a list of pointers to the potential reactant molecules in the system. So here, for this rule, we need a free scaffold A site, and we need a free A molecule as our second reactant. So the first pattern, the first reactant selector in our rule is an indexed list of the free binding sites of this type in the system, and we can see that there are something like 6. Then there are 4 free A molecules in the system. So that's our possible set of reactants, and 6 times 4 is 24. So the total propensity for reactions of this type in the system is going to be given by 24 times k plus 1, and so on and so forth for the other rules that are possible in the system. So at any given time, we know the total propensity for reaction events. We can select over the different rule types using standard Gillespie type algorithm. And then once we know which rule type we have, we can select instances of the reacting molecules here in a uniform procedure, because all the reacting molecules have the same propensity to react in this case. So we can, using a modified form of the Gillespie algorithm, then perform an event-driven simulation of the model. It's exact. We've avoided the problem of having to enumerate in advance all the possibilities. We'll instead discover the possible species as our simulation evolves in time. So now we have this other possibility. It took several years for the simulators to get good enough to handle arbitrary rule-based models. So we have, post about 2010, that was about when we finished work on first version of NFsim, the network-free simulator that works with BioNetGen. One of the network-free simulators, I should say, that works with BioNetGen. But anyway, we can use this direct method then to perform simulations of sort of rule-based models of arbitrary complexity, and we don't worry about how many possible states there are. We worry about basically how many reaction events we have to fire and how many trajectories we have to run to get good enough averages to do parameter estimation and so forth.
I'm sorry, you kind of lost me there on the last slide. So are you excluding certain configurations from happening? I'm not sure how you're cutting down the combinatorics.
We're not. We're just not. We're only considering. First of all, we're not indexing what species are present here. So there could be very, very many different types. We're only going to have to deal with. These are all instantiated as molecules in our simulation. So it's just, think about your section of computer memory. There's a very huge possible state space of different configurations that memory can have. But as long as I know how each portion affects the algorithm that I'm using at a given point, I don't have to know, and I have an algorithm for determining that. I don't have to enumerate all the possible states to figure out what's going to happen next. Maybe that wasn't the best way to explain it. Let me try again with our configuration here. So at each point in time, I have a finite list of molecules, right? And the only thing I'm keeping track of is what's bound to what. So I have my scaffold molecules in a list. Here are, the list. I haven't indexed them here for convenience. And then for each site in the scaffold molecule, I'll have a pointer that represents a bond, and it can point to another site on another molecule. And then there are sites for each of these A, B, and C molecules in lists. And all I have to keep track of is, at a given time, is what elements of my list have the capacity to serve as reactant 1 in my rule and serve as reactant 2. And I can, from that, determine the total rate of potential rule firings. Once I know, and then I use that rate to select over the possible rules to fire next in a simulation. So we're going to do an event-driven simulation. And really at no point do I have to do any algorithmic step that's more than the order of number of molecules in my system.
So I'm still missing something in that you clearly are showing different combinations of the species here.
Yeah, they can form. So I could, in principle, first of all, I can only sample, something that's order of the number of molecules I have in the system. But I don't need to. At no point do I try to enumerate all the possible different states that are here. Here there are 11, I think. But if I just simply let A, B, and C bind to any of the 3 sites, I could get, I think, some much larger number approaching 100. And at any given time, in this system, I wouldn't have anything near 100 different states represented in my system. Although over time, if I run my simulation, I would encounter probably all of them at some point. But I didn't have to enumerate them in advance.
Well,
yeah.
You enumerate them in advance? Or
yeah, it doesn't.
Demand.
You're fine. Okay. So the point is that so you only really get an advantage if the number of molecules that you're simulating is smaller than the possible state space. So here, if say I want to do a simulation of EGFR, I can simulate 100 EGFR molecules. The state space, remember, could be 10 to the 8. And I'm not going to probably encounter anything like 10 to the 8 in my simulation. The other flip side of that is that even when I'm doing the sampling, if I generate a reaction network that has 10 to the 8 or 10 to the 10 different possible events, at every step I have to select over those very, very large number of possible events. And I would need a very clever algorithm to do that, not to mention the memory to store all of those possibilities. So here I'm just looking in an algorithmic way at what can possibly interact as opposed to generating all the graphs of all the possible different interactions. This is I think I should move on in the interest of time. I recognize that there's often a conceptual difficulty here. And many people, it takes the whole network-free idea, it takes some time to sink in. And yeah, what are its relative advantages and disadvantages can be confusing. But the key point, which is somewhat, yeah, this is just another representation of the, I should say that I want to get to the key point in a second, but I should say that so this network-free simulator was developed by my collaborator, Thierry Emanet at Yale, and his student Michael Snedden in collaboration with me. And it was a really nice code that we continue to actively maintain and is part of basically the BioNetGen set of tools. Okay, so I wanted to mention that, but to go back now to how does this work in practice? So if we have a system that we're interested in modeling some kind of a receptor signaling system here, the IgE receptor, typically the way that it unfolds is we start building models of different levels of complexity. So here I'm representing contact maps for a bunch of different models where we've included different sets of molecules, different numbers of components, and they actually increase in the possible state space over time. And what's shown here is the computational cost per time step of running or actually for simulating a fixed amount of time, of running one of these, running these various models. So here I'm increasing the size, the potential network size of the model, and here I'm on the y-axis I'm looking at the computational cost. And what we see is the network-free method has a relatively gentle increase in computational cost as we increase the complexity of the model, whereas other simulation methods such as ODEs, for example, even the stochastic simulation increases fairly dramatically with system size to the point where somewhere in this region it starts to become quite difficult computationally, quite expensive to perform the simulations. And in fact, the stochastic curve ends here because the simulation for the network at this size didn't terminate in a reasonable amount of time. So the key point is that we can avoid this computational bottleneck in system complexity by using network-free methods. And it's really when, and what's interesting here, that this actual scaling curve kind of surprised me when we made it, is that the crossover between the network-free method, for example, and stochastic simulation or ODEs occurs at a size of network in this particular problem of about 10 to the 4 reactions, but that's a general, pretty common result that between networks of hundreds to thousands of species, we start to see that network-free becomes faster than other methods. It's earlier than we expect, and part of that might have to do with implementation issues, but the practical benefit of having a code like this is that you can build a model that includes the interactions that you want. I think this is really important for scalable modeling, and not really have to worry that you're not going to be able to simulate that model. You might not be able to do everything with the model that you wanted to do, but you can certainly simulate and see how it behaves and do some characterization of it. And recently, tools have been developed to do stochastic parameter estimation with these kinds of large rule-based models. And in fact, the T-cell receptor model that I showed earlier was fit the parameters of that model were fit to data using some kind of genetic algorithm-based approaches. So I think I view the network-free simulation technology as really an enabling technology for scalable simulations of these reaction networks, and I think it's particularly relevant in this context. So, okay. So the workflow looks something like this, and as I mentioned before, you'll do some of this in lab, so I'm going to skip past some of the details. There are a lot of tools that work with rule-based modeling, and I'm going to focus on one of them here, which pertains to how do we get reaction network models that have already been developed into a form that we can start to view them in terms of rules and the molecular components and interactions, and work with them in rule-based simulation tools. Okay, yeah, more diagrams. I wanted to mention that there's an active community of that's developing rule-based modeling methods. This is a very incomplete list, but BioNetGen and Kappa are the sort of two major languages that are most commonly used. BioNetGen, I think, tends to be used more in applications by people developing specific models of biochemical systems. Kappa has been very well studied in the computer science literature, and there's a proposed and nearly adopted standard for multi-component multi-state modeling in SPML level 3. That was largely developed by Martin Meyer-Schellersheim, who's the developer of Simian. Okay. So yeah, I've got more stuff here. This was a 45-minute talk last time I gave it, so but it just depends on the audience. So the point of this was just to tell you that there are active developments underway to develop more efficient simulation methods for rule-based models, and we're also trying to make spatial particle-based simulators so that you could model reaction diffusion in a cell in a for large-scale networks. Okay, and a summary: we can represent biochemical knowledge in a concise and precise way. The rule-based models are quite flexible with respect to simulation methods, so you can, once you have a model of the biochemistry, you've represented the knowledge of the biochemical system, you can simulate it in a variety of different ways. Potentially, there's the ability to generate reusable sets of rules, and there's been some work in this area already, and I think there's a lot more potential. And rule-based modeling opens up a number of paths to visualization, and that's what I'll talk about for the last 15 minutes or so. So we've already seen the contact map as a compact way to represent a fairly large network of interactions, and we'll see some other techniques as well. So probably many of you have engaged in what I call the standard modeling protocol, and it's the same for a rule-based model as it is for any other form of model, in terms of at least the first two steps. So typically, we identify what the components and interactions are in our system. We're going to need to go out and find rate constants or make estimates, or find some way to starting point for developing a simulation. And then, if we're making an ODE model especially, we typically somehow figure out a way to distill this into a set of differential equations. And often what happens is that something is lost in that translation. So we take our components and interactions and we make them into a network, typically a network of equations. This is an incomplete set. We don't have the V's, but the idea of the differential equations is conveyed here. So this is this is the most we get about models in the literature in terms of a visual representation. And then we typically get lists of equations. We might get an SPML file if we're lucky. Okay, but at this point, we lost the information up here. Okay, that's described qualitatively in the paper somewhere, but it's typically not in a machine-readable or visual format, except for this kind of structure, which is great, and it conveys information here, but it's pretty hard to tell, certainly not at a glance, what components have been modeled in this system. And I should mention, this is a relatively good diagram. Often we get almost nothing. So we want to know, what's in models, essentially, that have been developed. So finding what are the molecules and interactions that were used to build a given model, and what's the space of different molecules and components that have been considered in the literature. That's a really hard problem to solve. What are the assumptions that are being made by such models? And could we, if we could relax some of these assumptions, might that make it easier for us to join? Remember, going back to my idea of putting together different signaling pathways, could we join up models if we could relax some of those assumptions? So To address some of these questions, we developed a procedure that we call atomization. We convert this into a rule-based model, which we're representing here as a contact map. We find what are the underlying molecules and the interactions that are present in this model. So here's a model of the epidermal growth factor signaling pathway. We consider these molecular components and these binding interactions that are represented in the diagram, and then various sites of modification are also included in this model. So we have a tool, we can give it a reaction network, not a graph like this, that would be a pretty amazing tool, but we can take this model in SBML format and convert it into something like this. Jose Juan Tapia in my lab has been working on this for quite a while. This was a preliminary report on Atomizer, and we've extended it and improved the tool a lot since that original report. And the basic idea is we take stoichiometry of reactions. The naming conventions that are used in models can often be important. We have the capability to use annotation, but for all of the results that I'm going to show here, we've taken the annotation out in order to see what information Atomizer can recover. Okay, so in some cases, in the case of the models in BioModels, for example, BioModels database that we're going to, I don't have it here, BioModels database that we're going to use, some information has been provided to link some subset of the species in the model back to model databases, and we can actually use that information to test whether atomization correctly recovered the information that we wanted to get. So the idea, we have a reaction network model. We take the information about the stoichiometry and the names that have been given to the different species in the system, and we use that to determine what molecular interactions and modifications have been included in that model. And so the process looks something like this. We take our reaction network, we use our stoichiometry and lexical analysis to take our list of species and map that into a list of constituent species. So the EGF and EGFR molecules are just molecules, but the first complex that forms in the system, the EGF-EGFR complex, we can figure out from the first reaction that we see is comprised of EGF and EGFR. Then there's a dimer that forms that's composed of a complex of the two EGF-EGFR complexes, and so on and so forth. And we build up a table in our first pass that gives us the stoichiometry of species in terms of other species in our system. And we ultimately want to resolve that down into the basic building blocks. We can take our so-called species composition table and we then expand that. We do another pass through it to identify the stoichiometry of all of the complexes in the system, and also potentially the sites where molecules are being modified. And in some cases, the nomenclature by itself. So here's an example. We see a complex that the dash P clearly indicates that it was phosphorylated. It's a phosphorylated form of this earlier complex that was encountered. But Atomizer doesn't know a priori where that modification took place. So it should be on one of the molecules, but we don't know which one. And so here, the user is going to have to be queried to provide that information, unless we have some, potentially we could use annotation for this complex to say that it contains phosphorylated EGFR, but we very seldom get that kind of information. So here we would have to ask a user to say, okay, your EGF-EGFR complex is phosphorylated, tell me which site it's phosphorylated on, or how to describe that complex. Once we have this basically table of species stoichiometries, we use that to generate a species, the set of graphs that represent all of the species. So how are these guys connected to each other? That can be an involved process, and in some cases it will involve, so to find the binding, for example, in the EGFR dimer here, we know from the model that EGF binds to EGFR, but we actually don't know in the case of this complex when the dimer forms whether the dimerization involves receptor-receptor interactions or ligand-ligand interactions, and either possibility is present. So we actually go out to an external database and based on the queries that we perform there, find out the most likely site of the interactions, and then we put that back in. We can alert the user that that information has been used, and any decision that Atomizer makes can be overridden in this process. But the goal, we're really trying to automate it as much as possible so that somebody doesn't have to go through an exhaustive process in order to get a very good approximation of what the original assumptions were. Okay. Okay, so we can do sort of what I think are kind of magical things, like take a very complicated model which has hundreds of reactions, and figure out at a glance what the molecular components of that model were. And we can look at in detail at any of the reactions in the model. We can look in detail at any of the molecules in the system and see what kinds of modifications steps they undergo. We can also easily compare models to each other. So here's the Sherbel model of EGFR, and here's the earlier Holodenko model of EGFR, of the EGFR system. And we can see that there's a very high degree of overlap in these early components. They're basically modeled in the same way in the two different models. Holodenko considered PLC gamma. That was left out of the Sherbel model, which then extended the whole pathway to consider the MAP kinase activation. And we can, let's see, we've applied this to BioModels, and what's shown here is basically the percentage of the reactions in the system that we can get atomization information out of. And it's sort of this bar indicates models that can be completely atomized. Here there's some varying degree of information that we can extract, typically a lot. And then here are sort of an intractable set of models that are specified in a certain way in BioModels, where there are actually no reactions in most of these. And so we would have to go and analyze directly the direct encoding of differential equations that's been done in this subset of models. So there's an intractable core, but the vast majority of models we can extract structure information from. We've done a lot of validation to make sure that the annotation that is in BioModels agrees with the stoichiometries that are recovered by Atomizer. And probably a more relevant measure of our success is that this now looks at the number of, the amount of input that the user has to give during the atomization process. So the majority of models in BioModels that we encounter, we can atomize fully without requesting any additional information from the user. There are some models that require a lot of information, and a fair number of models where there are queries of a few queries that have to be resolved. And we can look at, based on the fact that many complex species are represented with an underscore somewhere in the label, we can find out how much work is left undone by the atomization, and we see that for the most part we recover almost all of the, we get sort of full stoichiometry information on all the species that are present. There are definitely some issues that come up there. So we can start to do something like a comparative analysis of many different models that include EGFR. It doesn't project so great here, but each of the rows represents a contact map for a model, and then the green part represents an overlap with other models that are in the BioModels database, and we can start to see which models, what components have been modeled in which different models. Something else that I think is really useful that we can start to do too is to drill down and look in detail at how different molecules in these models have been modeled. So like the EGFR receptor, for example, has been modeled in the Sherbel model, has 8 different component sites to represent different properties. This diagram is hard to walk you through in a short period of time, and I'm running out of time, so I won't go into the details. But each node here represents a different possible state of the receptor, and each process, each edge, represents a different single site modification that occurs. And so just the graph structure overall indicates something about the way that this molecule has been modeled, and we could then use that to compare, for example, the way the same model in different same molecule in different models has been modeled. So we see here the GP130 molecule that's present in a number of the models that also deal with components of the EGFR system. That is, we can see how it's been modeled in two different models that are in the database, and we can see that up to a certain point, it's been considered in exactly the same way. They have the same set of binding sites, for example, but the binding interactions for one of these sites has been extended in 151 compared to 543, and it's sort of it's a strange process. So here you can either bind SOX3, STAT3, or SHP2 to this site. I forgot why. Oh, I forgot why there's a tilde there. But anyway, a caret there. But at any rate, what the key point here is that there's all this extra stuff that's been considered, and there's a cycle where all of the STAT3 and SHP2 and SOX3 are allowed to bind all at once if the binding is sequential, and then it's all reversed in a single, possibly reversed in a single step. So, what the significance of those choices are is not immediately clear, but the fact that different choices were made in these two different models and could account for differences in their behavior, I think, is really key. And it's hard to get this kind of information without really digging into the guts of the model. Another thing that we can do is compare how the parameters that were used to describe different processes in the two models. So here is a model of p53, and the parameters governing phosphorylation and MDM2 binding are somewhat different in the two models, and we can see those differences at a glance. Okay, so I have another module here, but we're running down to 4 or 5 minutes left. So, okay, I'll say a few words then about visualization. So we've talked already about I've shown you a number of different visual representations that zoom in at different levels of the model of models. And one of the problems that we had it's been ongoing with rule-based models is that although we have a nice graphical overview in terms of the contact map, so we can see what the components of the model are. And here I'm showing a model of an autophagy network that was recently published. Underlying that model, of course, is a lot of code. And the rule-based languages allow us to keep track of, write sort of what I call locally comprehensible rules. But when you start to assemble a large set of these interactions, and you want to know something about how they all fit together, it can be quite a challenge. So we wanted to find global ways to look at a set of rules that could tell us about the flow of information in signaling networks, and that would scale to large rule sets, so possibly models containing hundreds of rules. So why do we need rule visualization? So when we're debugging a model, it's very helpful to see how the rules fit together and if things are working as we intended. If we want to share models with other people, they need to be able to understand our models, and not everybody is going to want to spend the required time to fully digest a large block of code like this that's very dense. Our biological collaborators definitely don't want to delve into this realm. And then, as I've shown, comparing different models at the level of doing a code analysis like this is really pretty much impossible. And ultimately, that affects our ability to integrate different models together to make unified models. So one of the first things that we worked out how to do was just a simple way to translate this kind of a rule into a graph that could be compactly visualized. So up to now, I showed you rules that were framed in terms of a simple reaction notation, A plus B goes to C. Here I've compacted that into a single step, so we have two different patterns that represent reactants in a rule, and then the transformation that's applied to them is just to create a bond between two components. So this just zooms in on exactly the action that's carried out by the rule without making us look at two different copies and have to do a visual mapping. We can then chop up this this visualization of a single rule. So this is an exact representation of the underlying code. We can now coarse grain this a bit, so chop this up into its various components to make nodes for each of the sites that were in the patterns in this rule, and then a different node type for the rule itself. So we're going to make a bipartite graph on sites and rules that enable us to see. So the dark arrows are going to indicate the sites that are modified by a given rule. The lighter arrows are going to indicate the sites that are required to be in a particular form in order for that rule to fire. So this seems like it's not maybe very clear from this picture that I that I did anything that was going to make this kind of visualization more compact. But now I can aggregate these graphs over a large set of rules and make a unified graph, which unfortunately doesn't project very well on a screen of this size. But we can take our full set of rules and make a quite navigable network of the rules and the sites that they interact with. And if we lay this out in a clever way, and I'll show you some improved layouts in just a sec, we can actually do a nice visual analysis of this network. So just to be able to go from here to here in a precise way was a big step for us. And then clearly we have a large number of we have a large node node link diagram here. And we're going to be confronted with all the usual graph visualization problems of scalability and layout, but we have some powerful tools for dealing with that. So we can very easily remove what we call background nodes from this diagram and get something which is becomes very comprehensible, starts to become very comprehensible in terms of, for example, the percolation of phosphorylation and binding through the network. We can further group nodes according to shared properties. For example, the activating phosphorylation sites on given molecule can be aggregated together. And at the end of the day, we can collapse that all back down together and make a relatively simple diagram for our model that corresponds to the diagram that was used initially, sort of as an overview of the architecture of this network. We can generate that automatically from the underlying rules in a principled, in a precisely defined way. And so each of these steps we've described formally and provided tools that enable someone to carry them out. So you can document, you can make a graph that visualizes your model, and then you can make a collapsed graph that visualizes your model using a precise and recordable set of procedures to obtain that graph. And we can just see by visual comparison here that we've essentially recapitulated a hand-drawn diagram of this architecture, but in a principled way from the underlying quite complex set of rules that was used to describe all of these processes. So we would like to be able to basically go back, if you think about this as zooming, this is a very high-level view, and we can zoom down in to the view of individual sites in the network and see how they all interact with each other. So again, I think really important tools for developing scalable models. Here's a library of models of early events in IgE receptor signaling. So here we're going to have many of the different molecular components of such signaling pathways. And many different rules, and we could take subsets of these rules to develop, for example, models of particular systems. But we need to be able to see what's in our library, and so this kind of visualization system can be used to do that in an efficient way. So these are the topics that I covered today, and I think as I said, we'll get to see examples of how rule-based models are specified and simulated in the lab. And for those of you who are interested, I'm happy to show you how to use the Atomizer and the visualization tools that I've described in the second part of the talk. I should just thank the people in my lab who did a lot of this work. You see Sanjana, who's here. John Sikar worked on the visualization part that I showed at the end. Jose Juan developed Atomizer and also worked with John on a lot of the visualization elements. And everyone in the lab has contributed code to the projects. And we have a number of both computational and experimental collaborators with whom we work regularly. So with that, I'll thank you and be happy to answer any more questions you have. So I think.
You mentioned parameter estimation once or twice. Are there any tools or any secrets how you do it?
So we have, yeah, there are now a growing number of tools for parameter estimation. So you can, one thing that can be done relatively easily, I didn't emphasize it here, is to export the models in whatever format you like, so SBML or MATLAB or MEX code, for example, so that you can compile the model into C code, and then you can use, whatever parameter estimation tools are out there. We've also developed some of our own. So we have a Bayesian Monte Carlo parameter estimation tool that's called P-Tempest, and we use that in-house to do most of our work. And the basic idea is that you do MCMC, but we use multiple different temperature chains to accelerate the convergence of the process. So that's one possibility. And my collaborator Bill Hlavicek, his group developed a tool called BioNetFit that was just published, I think, the end of last year, that lets you do. I think you use so far they've coded genetic algorithms, but they're actually very actively working on that tool to develop many different methods. But the nice thing that it does is it lets you run NFsim on as many cores as you have in your system, and then do parameter estimation on stochastic models. So I think it's not the algorithms are being developed and being used by many different groups, but having the infrastructure to do the work in an easily accessible way is pretty nice. So the idea of BioNetFit is that you can put it on a cluster and it sort of figures out how to run the jobs on the cluster without the user having to know a lot of details about how it works. So that's kind of one of the directions that it's going in. Yep.
So you talked in the beginning about the combinatorial explosion of states. So is there evidence that the approach you're using has been validated experimentally, that you can kind of weave your way through that combinatorial explosion to find which states are actually happening biologically at what rates? Is there experimental validation of these models, of the methodology?
Yeah. That our study sections love that question. Yes and no. I mean, there certainly have been discoveries that were made because we built models that included mechanisms that hadn't previously been considered. Does that mean that the system had to use 1,000 different phosphorylation states to obtain that? Again, there is a big problem that it's very difficult to observe the complexity in the system itself. We don't have ways to look at in one protein this phosphorylation status of more than a couple of sites at once. To the extent that people have done it, they see all kinds of crazy stuff. So I invite you to look at, there's a paper from Seth Grant's group in Science Signaling from 2009 that comes to mind, where they looked at the and this is true for many phosphoproteomic experiments that have been done. There's all kinds of things that get phosphorylated in these networks. So there's almost no control, over the flow of information that's evident when you look at the data, at least at first glance. So I think that I could flip the question around on you. Is there any evidence that the system works as simply as most people try to model it? And the answer is no. There's very little evidence that it works as simply, except that the models sometimes work. So I usually start off with something, some philosophical statement about modeling, something like, all models are wrong and some models are useful. And I think, everybody who does modeling in this field is very well aware of that, your model is wrong. It's not a precise representation of reality, but to the extent that it gives you insight into things that you otherwise wouldn't have thought of, it can be useful. Yeah.
I guess I'd suggest there are two possibilities biologically. One is that biological systems really can explore all the different states that exist.
Right.
The other is that because of some of the simplifications that I suggested early on about exclusion and fast rates for certain interactions, that the biological system actually is constrained to a relatively small number. Now, we may not be
no, we don't. Yeah, I agree with you, but I agree with that there's a lot of constraint. We just don't know what the constraints are, and so we need we need principled ways to find them. Actually, I have a set of slides, but I'm not going to, don't worry. I'm happy to talk to you about that offline, because that's something that I think it really needs to be done, and we have some clever ways to approach that topic, actually. Yeah, in the back.
But it seems like to deal with the dimensionality problem of combinatorics, right? I mean, you took the path that any computational person would take, even Monte Carlo simulation and dynamic programming, or even other kind of a Turvy algorithm, where you get to a place where you have something, and then you look iteratively to say, well, where am I going next? And by doing that, you reduce the set of possibilities that you have to investigate at each iteration.
Right.
On the other hand, if you're sampling such a small portion of the space, what's the evidence that you're sampling the right portion, or that you're just ignoring a huge fraction of the space that is meaningful? And if you're doing that, I'm not sure how valuable the model is if you're ignoring a huge fraction of the space. And it's a...
We're not, so to be clear, network-free simulation is a completely equivalent way of simulating the system to doing standard Gillespie. It's exact. There's no approximation being made there. We're just controlling the way that we explore the state space by simulating a relatively small number of molecules. If we simulated, 10 to the 23rd molecules, we would have all the states in there. But so it's not an approximate method. We haven't, the where you would make approximations is in how you construct the rules. And the way that we've constructed the rules is, in a way, very approximate, because we've assumed that there's very little interaction of the form one site affects another site. So the canonical, the standard assumption that people make to make these models simple is that basically each molecule can only do about one or two things at a time. If a site gets if a EGFR gets phosphorylated, it's phosphorylated, it can bind one adapter. Okay, even though it's in a complex, it might be an oligomer with 4 or 5 EGFR molecules, it's just going to bind one adapter. So basically you're just coarse-graining the stoichiometry of complexes. So here, the rule-based approach starts from the point of view of, I'm looking at what sites can interact with what other sites, and if an interaction is there, then it's not affected by anything else that's going on in that molecule. So a given molecule that can interact with 5 things could in principle interact with all 5 at a time, and I'm not going to restrict that for my system. For sure it is. The biology restricts it, but we don't know the rules for the restriction. And when we know those, we can put them in.
You mentioned the parameter estimation. Which type of data do you fit your model on?
Whatever data we can get. Most of our projects, we deal with population data like Western blots, some flow cytometry. If we can get single cell data, Sanjana is working a lot, for example, with single cell trajectories. But we have to, there's a trade-off. You want more measurements of more things. You want them to be time resolved. Ideally, you would like them to be time resolved inside of a cell, inside of a single cell. But that's a topic for a whole other talk. Yep.
That issue we were just discussing about the constraining the complexity, the combinatorial complexity, by limiting the number of molecules in your simulation. As we were discussing, each time you do a simulation, you explore different routes transduce the information from receptor to nucleus. So I guess that's what happens in a cell as well, because there are small numbers of these molecules in cells. So the cell is exploring different, if every interaction then is as likely as one another, then the cell explores different routes through to transduce the information each time too.
Yeah.
So in that sense, what you're doing is quite biological. Is there lots of noise associated with simulations from run to run to run when you have a very large state space? In other words, does it matter if you navigate your way in different routes through the network?
There have been a few studies of that. In terms of, yeah, I don't have a good answer for that.
I was thinking about the rapidity of the transduction of a signal.
No.
The rapidity
How much variability is there from intrinsic noise would be one way to frame that question. And it really depends a lot on the model, but typical signal transduction models with the mechanisms that we've considered up to this point, no, there's not a lot of intrinsic noise. Their copy numbers are high enough, tens of thousands of molecules, that you don't see a lot of variability from run to run. If you put feedback into that, you could easily change the story. When you get to transcriptional level, you can definitely change the story, but there are not so many models that go out and in and back out again. And I think when you start to do that, you see more variability, but not when you're worried about things that are happening in the early events and signaling like we mostly have focused on so far.